Okay. Who can tell me something that is inside, comprises a photosystem? One thing, Maddie.
Pigments.
Pigments. All right. So, what's the main pigment in photosynthesis? It's the main pigment in photosynthesis. Charlotte. Chlorophyll, and specifically chlorophyll A. So chlorophyll A is right here. I'm going to put here, so this is chlorophyll A. I'll just put a little A for chlorophyll A. Is chlorophyll A the only pigment? No, there are other pigments. Chlorophyll A is the main pigment in the center part of the photosystem. They call that center part, you remember? It's called the reaction center. And chlorophyll A is part of that. But there are other pigments around here. So I'm going to put these as little dots here. And remember that all pigments in general, their job is to absorb light energy. So I'm going to draw here in a different color here, light coming in. So this is light being absorbed, and this happens simultaneously with both of these photosystems. So light's coming in simultaneously, being absorbed by the pigments. Remember though that that light energy goes from pigment molecule to pigment molecule, but eventually ends up in this chlorophyll A molecule. So the chlorophyll A gets all of the light energy eventually. So why is it that we can't just have in a plant cell chlorophyll A? What's the benefit of having all these other types of pigment molecules? They call them accessory pigments. So they're helpful pigments in photosynthesis, but they're not the main one. Miranda?
They absorb like different wavelengths of light.
That's right. They absorb different wavelengths of light so that when white light shines, that white light is the whole color, all the nanometers of visible light, the Roy G. Biv of the spectrum, that if you had just chlorophyll A, it would only absorb one particular nanometer of light, but having multiple pigments or absorbing different wavelengths of light allows the plant to absorb that much more energy. And so then it eventually gets transferred to chlorophyll A. Can somebody tell me what happens to chlorophyll A when the energy is absorbed by it? Noticing posing a lot of different questions. I want you to really focus yourself on paying attention to this and seeing if you know the answers to these. That's a kind of a test for you. And so if you don't know the answers to these, then you either come in on Thursday for help or spend some time studying. There's no homework tonight, so you can spend your time studying, all right? But I really want you to focus on trying to see if you can answer them, whether or not you raise your hand and answer them verbally, but I'd like you to try in your head. All right? So what happens to chlorophyll A when the light energy is absorbed? I'm going to call on some random people, because I know Barack is not the only one in the class that knows. I appreciate you raising your hand, but I want to see some other people. What do you think, Izzy? It's okay if you don't know.
Okay, so what was the question? What happens to
chlorophyll? So when chlorophyll A, that light energy is absorbed by chlorophyll A, what happens to it?
Does it have to do with the electrons being excited and they like jump the level?
That's exactly right. The electrons get excited, all right? So 2 electrons, I'm going to draw here in blue, 2 electrons here, get excited. And they get excited, but then when they become not excited anymore, they don't drop back down to chlorophyll. What grabs them?
The primary electron acceptor.
Yeah, there is a molecule, I'll put PEA here, the primary electron acceptor, that grabs those electrons, all right? So that's the name, it's a primary electron acceptor, accepts the electrons. But now we have 2 chlorophyll molecules that are missing electrons. And guess what? The chlorophyll can't work anymore if it's missing 2 electrons, so it won't be able to do any more photosynthesis. So therefore, we have to replace those electrons. So let's talk about this one right here. This chlorophyll A molecule just lost 2 electrons. They got excited, and now they're with the primary electron acceptor. Bless you. Where do these electrons that got excited here, how are they replaced? We need to put more electrons in their place. What replaces those electrons?
Is it the electrons from the
Yes, the electrons from the water. So water is used here, and I'll put I'm going to do all my electrons here in blue. So the electrons come from the H's. All right. So the 2 electrons from the 2 hydrogen replace those electrons that were excited here. That leaves behind, when we use the water, that leaves behind 2 H pluses. Because the electrons part of hydrogen is now here, and that leaves the H pluses, and they write it as a half of an O2 molecule. That's one O molecule, one O atom. They just don't write O because technically, in nature, one oxygen atom is not ever found. It's diatomic, so that's why they write it that way. All right, so therefore that's the replacement of those electrons. Let me ask you this, what replaces the electrons in this chlorophyll molecule? When these electrons get excited, this is missing electrons. Where do these electrons come from that replace the missing 2 electrons? Where do they come from? Maddie?
The ones from photosystem 2 travel through the electron transport chain.
That's correct. So these electrons now are the primary electron acceptor, and then they go through the electron transport chain and end up with chlorophyll A. And so they move through the electron transport chain. Why do they move through the electron transport chain? For what reason do they move? What's the reasoning? Miranda?
They move to create energy to pump the H plus ion out or into the cell.
Okay, so that's what happens when it moves through here, is it pumps H plus ions where?
Into the cell.
Into there. All right, so H pluses go from here to here. Now we're using the word pumped. What does that imply about the situation? If it's pumped. See?
It means it's going against the concentration gradient.
It's going against the concentration gradient. Pump means that it uses energy. And so therefore, where's my higher H plus concentration? In this space here. What is this space called? The stroma, right? Is it here or here? Inside. So it's going to go from low H plus concentration to a high H plus concentration. Hence the need for energy. And the energy comes from these redox reactions where the electrons flow from one molecule to another. They get reduced and oxidized, reduced and oxidized. That releases energy. That's the energy used to pump the H pluses. Why do they go from one molecule to the other? Because each one is more electronegative. That's why they move down the electron transport chain to begin with, and that's a good thing because it pumps the H pluses in. And so we'll come back to these H pluses in just a second. Going back over here to photosystem number 1, now we have 2 electrons with the primary electron acceptor. What happens to them next? And actually, this picture from this does not go into the detail that we did in our notes. Does anybody know what happens to those 2 electrons? I'm going to pull up the picture from our notes too. Brock, you know, you can say it out loud while I'm looking for the picture.
They travel through another small electron transfer chain and go into NADP plus reductase and then the NADP plus plus the 2 H plus ions form NADPH and an extra H plus ion.
That's correct. All right, and this is what this picture is talking about. Shows. This is from our notes. So here we are at photosystem number 1. All right, photosystem number 1. So there's a small other little electron transport chain comprised of, in part by an enzyme that's going to help make NADPH. So I'm going to go ahead and draw those in here. All right, and then show that my electrons are going to went up to the primary electron acceptor, now coming through here, and is going to meet up with NADP plus 2H pluses, and make NADPH. And this is one of the main things we want to get out of the light reactions. All right, so that's that. So why does the cell expending energy to pump H pluses inside the thylakoid? Why is that important? Why is H pluses inside the thylakoid important? What do you think? Andrew, do you know? So why is it important to pump H pluses inside of here? What are these H pluses going to do? What's the importance of them? They're going to be pumped. Are they going to be pumped or diffused? They diffuse. And they diffuse through an enzyme. Somebody else want to help them out? There's an enzyme, Jace?
They go through ATP synthase.
That's right. It goes through ATP synthase. So I'm going to go ahead and draw ATP synthase here. So this is ATP synthase. These same H pluses, I put in red here, are going to diffuse. Why is it diffusion? High to low concentration, so it doesn't require any energy. When the H pluses diffuse through ATP synthase, what does it do to the ATP synthase?
It puts it in its active state.
Yeah, and so it changes that shape so that what two things can bind to the active site? What would be the substrates? Manny? ADP and a phosphate. And then they will bond and make ATP, which is your second major thing that we want to get out of the light reactions. All right? And so those are the light reactions in a nutshell. All right? So with that being said, let's go back to the packet and go over the answers to the questions dealing with the light reactions. So let me put the picture back up from the packet here. These are the light reactions. All right, so number 8 asks you what shape or symbol represents a single electron. These are these small dots here. All right, this is the path of the electrons. Number 9, find 2 places where electrons are released from chlorophyll. So that would be here in photosystem 2 and here in photosystem 1, where the electrons get excited and leave. And go to the electron transport chain. B asks you find a place where electrons are released from water. You should have here, it's by photosystem 2 in the inner, inside of the thylakoid here. It has to be inside the thylakoid because when water is broken, you get H pluses that contribute to the hydrogen ion concentration. This high hydrogen ion concentration in part is due to those H pluses from water. Then C, when electrons are released from water, what other products are formed? That is your O2 and your H pluses. All right, turning the page. Number 10 asks you to put the letters A, B, and C based upon where the things above are occurring on this picture. So excited electrons leave chlorophyll and reduce NADP+. You should have an A in this area here, all right? Because that's where that's happening by photosystem 1. For B, with the excited electrons moving through the electron transport chain, the publisher put the B here. I think the B should be more here where the electron transport chain is, where the H pluses are being pumped. And then C, the hydrogen ions flowing through ATP synthase. That's right here, making ATP. So this is your ATP synthase right here. Number 11 asks you to describe how the system, the electron transport chain system works and what job it performs. So the electron transport chain, as we talked about, uses the energy released from the electrons as they move from one molecule to another. Since it's a redox reaction, energy is released. That energy is used to pump H pluses into the thylakoid, and so that's the purpose of it. That's the job, is to pump H pluses into the thylakoid. Number 12, the embedded protein complex found in the thylakoid membrane that uses excited electrons to produce NADPH, and that is photosystem 1. 12B, the one that splits the water, that would be photosystem 2. And the protein where the H plus is diffused to make ATP would be ATP synthase. All right, so 13, turning the page. What's the source of replacement electrons for those released from photosystem 1? So here is photosystem 1, And so the electrons here that were replaced when they got excited came from the electron transport chain. Then B asked you about photosystem 2. These electrons were replaced from water. 14, is CO2 involved at all in this? Have we used CO2 at all? No. So CO2 comes in the next step. 15 asks you to write a chemical reaction that summarizes all of the chemical reactions in the light dependent reactions of photosynthesis, starting with 2 water molecules. So they're asking you to look at this picture and write everything that comes into it. So what are the reactants? So we need 2 waters. We have here 3 ADPs plus 3 phosphates to make your ATP. And we have 2 NADP pluses plus the 2 H pluses here. So those are the things that go in. And so then you'd have an arrow, so the things that would be on the left side, and then an arrow, and what is the products? What do we get at the end? O2, 3 ATPs, 2 NADPHs. All right, that's the other side. So then it says 12 water molecules are shown as reactants. So in the equation, 12 are going in, but 6 water molecules are shown as products. Are any of these water molecules products of the light-dependent reaction? Is there water coming out of any of this? No. All right, only water going in, so the water comes out of the Calvin cycle. And then calculate the total number of oxygen, NADPH, and ATP molecules that are produced when 12 water molecules complete the light-dependent reactions. And so here it's showing you with 2 water molecules how much is produced: 1 O2, 3 ATPs, and 2 NADPHs. They want to know about 12 water molecules. So we'd have to take our numbers and multiply them by 6. All right? So if we have 12 water molecules, we get 6 O2s, we get 12 NADPHs, and we get 18 ATPs. Okay? And then where do they go after they're made? They go to the Calvin cycle, which is the next step. All right? So let's look at that. And let's add that to the board here, and then we'll go over the answers to those. All right, so this is the Calvin cycle, or light-independent reactions. So let's add these to our board here. So the NADPH and the ATP are going to go to the Calvin cycle. So here we have the Calvin cycle, and it's a cycle, meaning that the molecules that we start out with we're going to end up with, and so it can go on and on and on. So what comes in here is a reactant. This is where our CO2 comes in. And so you have CO2. Now notice here, let's show this picture here. They have 3 CO2s coming in, and then they have 6 of these, 3 of these. In our notes, I talked about in real life the CO2s come in one at a time. So therefore, to make one P-gal that comes out here, you need 3 CO2s, but they come in at one at a time. So in our textbook, in our book, they talk about how there's 3 turns to make one P-gal. This publisher has chosen, because since you have 3 CO2s total, they tripled everything in this to get your 1 P-gal. All right, so I just wanted to point that out. It's the same idea, it's just whether or not you show it. I will not ask you how many terms of the Calvin cycle, okay? It's just the overall big picture here. So, I just wanted to point that out here. So let's look at this. They just subdivided this into 3 phases, and we did this in our notes as well, but I just want to make sure that everybody understands this carbon fixation, so C fixation here. So let's look at what happens with that. This is where your CO2 comes in. Notice that you have a line here and a line here with this arrow. What is this telling you? If you saw that, if it was something you've never seen before, what is this telling you right here? See?
That CO2 is being added into the reaction, so it's like not there at first, and it has to be added in order for it to keep going.
Okay, yep. CO2 is added to the reaction. When these guys come together from the RUBP and the CO2 and go to PGA, what can we assume about these two? That this is a chemical reaction. These two react, and after this reaction, you no longer have CO2 and you no longer have RUBP, you now have PGA. All right, so they've reacted and formed new molecules. this process here is called carbon fixation. In general, I talked about this in the notes, a general definition of what carbon fixation means. Anybody have an idea? Jace?
Isn't that when carbon is added to an organic
To an
molecule?
organic molecule, that's right. So carbon is added to an organic molecule. So you have CO2 in the atmosphere. Now that carbon that was once in CO2 is part of an organic molecule inside of a living cell that it can do stuff with, all right? And so that's called carbon fixation. Whenever you see fixation, later on in the course we're going to be doing an ecology unit, and we'll talk about nitrogen fixation. So like for instance, there's nitrogen in the air that we breathe. You're breathing in nitrogen all the time. You can't use nitrogen in the air. It's N2 in the air. It's not usable, but yet we need nitrogen to make proteins and so on to survive. So we need nitrogen. So therefore we have to eat our nitrogen. But that means something on the earth has to be able to get the nitrogen from the atmosphere and put it into their body so that we can eat it to have nitrogen. And so we'll learn that there are what are called nitrogen-fixing bacteria in the soil. They can take nitrogen from the air, put it into an organic compound, and then other organisms eat it, and so on and so forth, and then we can get nitrogen. All right? So fixation means that it's incorporated into an organic compound. This happens to be carbon being incorporated into an organic compound. So that's your carbon fixation. That's the first step. And so then we have the reduction phase. What happens during the process of reduction? And be more specific than something is being reduced. Charlotte? Electrons are added, absolutely. So what's going to happen here is notice that these 6 PGAs are going to be converted to 6 PGALs. All right, and so in order to convert that from here to here, this is a chemical reaction. There's different enzymes that are involved here that we don't need to know about. Some energy is needed for that conversion. And also notice that we use NADPHs. And so what happens is, this is called reduction, so that this is getting electrons, is being reduced, electrons are added. Those electrons come from NADPH. Remember, NADPH are electron carriers, and so they provide the electrons, and that's why it's reduced. All right, so you have that reduction phase. After the reduction phase, out comes a pGal molecule, right? So here's your pGal. How many carbons big is a pGal molecule, do you remember? 3 carbons. So now notice that 1 carbon pGal comes out, and the rest of them stay in the cycle. So the rest of these, what are they going to do? They're going to be converted back to RUBP so that the cycle can continue to happen, so that more CO2s can react with RUP and the cycle can go on. Which is why this is the regeneration phase. What are we regenerating? RUBP. All right. So that's your regeneration phase. And so, let me ask you this: how many pGals does it take to make a glucose? How many pGals does it take to make a glucose molecule? What do you think, Joe? 2. And so when I say pGal is a 3-carbon molecule, if I multiply this by 2, I can get a glucose molecule, because glucose is a 6-carbon molecule, right? And so that's why we say this whole process would have to happen all over again. And so to get 2 of those, so another 3 CO2s to get another pGal, that's why there are 6 CO2s going into photosynthesis, all right? And so when we do this, though, this fixation and reduction and regeneration, one thing I didn't add on to this is it uses the ATP and the NADPH from the light reaction. So when I drew the arrows coming in here, we can draw them here where you have the 6 ATPs I used here, just like I did here, where you get 6 ADPs, and you get your NADPHs being used as well, becoming NADP+, all right? And then we can add here the ATPs. ATP. So on your picture, you could add those here, all right? And then when they break down the ATP, where do the ADPs go? These guys all go back and are reused in the light reaction. So this, this, all this goes back and used here. All right, back to the light reaction. So they're reused, they're recycled over and over again. All right, and that is the Calvin cycle. So let's go over the answers to these. So for 17, hopefully you had carbon fixation, a reduction, and regeneration. 18, how many RUBP molecules are used in one turn? They're talking about one combined turn to make one P-gal, so it's 3 CO2, so they're talking 3 RUBPs. B, how many carbon atoms are in each of these? If you counted them up, you should have gotten 5. And then it asks you how many total carbon atoms are in these 3 RUBPs. One has 5 carbons, so you multiply it by 3 and you get 15. 19, RUBP combines with CO2 to make PGA. A, how many CO2s do they use to do that? 3. B, how many PGA molecules are made? 6. C, how many carbon atoms are in a PGA? Only 3. But how many total carbon atoms are there in these 6 molecules? Each one has 3. You multiply by 6, so you get 18. They're trying to show you here conservation of matter. That matter, the matter can be neither created nor destroyed. We had 15 carbon atoms here, 5 in each RUBP, times 3, so we had 15 here. Add an additional 3 from CO2, so now we have 18. We should have 18 carbons there. Question?
I have a quick question. Just in the notes, I noticed that P-gal was called P320.
It's the same thing, yes. There are 2 ways to say the same thing. Yeah. That's a good question. That was the same thing in cellular respiration. We had that too. All right, yeah. What number are we on? 20. What happens to the carbon atom from carbon dioxide? It is now in PGA. And then 21 is asking you about carbon fixation. We already talked about that, where the carbon becomes part of an organic molecule. All right, so that is the carbon fixation stage. Now we're going to look at reduction here. So what molecule does the PGA turn into? And it turns into P-gal. All right? And then 6, the PGA, what happens to it when it converts from PGA to P-gal? If you look here, it has an oxygen atom right here, no oxygen atom right here. So it has an H instead of an oxygen atom. So it lost an oxygen atom, gained a hydrogen. Identify, see the types and number of molecules that provide the free energy to reduce the PGA, and that is your ATP and your NADPH. And then the total number of carbon atoms, has it changed? We have 18 here, and you have 3 carbon molecules here, so we have 18 and 18, so no, it has not. So then, next page, 23, water is a product. Right here, here's the water, and it says how many water molecules are produced, and that is 6. B, explain where the hydrogen and oxygen atoms in these water molecules originated. So how are these water molecules made? The oxygen came from getting rid of the oxygen of the PGA from here to here. So that's where the oxygen comes from, and the H's come from NADPH. 24, how many P-gal molecules continue on to the regeneration phase? That's 5. Remember, 1 comes out. A and B, identify the number and types of molecules that provide the energy for the regeneration phase, and that's the 3 ATP. C, how many total carbon atoms remain in the Calvin cycle at this point? So after there were 18 here, you lost 3 in those 3 carbon molecules, so now you have 15 left over. And what molecules regenerated? I already posed that question. It's the RUBP. How many total atoms leave the cycle before the regeneration phase? That's the 3 in the P-gal. And what happens to the P-gal molecule that does not continue? It's used to make glucose. All right, that's what it's used to. Remember, 2 of them make a glucose molecule. All right, so then next page, 25. How many P-gal molecules will it take to make 1 molecule of glucose? I already answered this. It is 2, remember, because they're each 3 carbons long. B, how many turns of the Calvin cycle will it take to make 1 molecule of glucose? And this, where they put 3 as once and tripled everything, it's 2 turns as it's written here. Realistically, it's 6 turns because each CO2 comes in by itself, all right? But how it's written here would be 2. And then C, calculate the total number of ATP and NADPH molecules used in the production of 1 glucose. So in this case here, all of these totals are for 1 p-gal. You need 2 p-gals to make a glucose, so therefore we have to multiply everything by 2. So there's 6 ATP plus 3 ATP is 9 times 2, so we'd have 18 ATP. And 6 NADPHs times 2 would be 12. All right, so 18 and 12. 26, where do the ADP and the NADP plus go? They go back to the light reactions. So they're reused in the light reactions. And step 27, how do they depend on each other? Light and reactions in the Calvin cycle. So the light reactions provide ATP and NADPH to power the Calvin cycle. And then the Calvin cycle, those ADPs and NAD pluses are reused again to remake those, so therefore they work together in recycling those. And 28, you had to put underneath it where these occur. So for 28, for the 6 CO2s underneath it, you should have they're used in the Calvin cycle. The 12 H2Os are used in the light-dependent reactions or light reactions. Then on the other side, the products that are produced, it's not used, so it's produced in the Calvin cycle, the glucoses. 6 O2s are produced in the light-dependent reactions or light reactions, and the waters are produced in the Calvin cycle. 29, you had to put these in order for the flow of energy. So it should be, start out with sunlight, then they're in the high energy electrons, which is used eventually to make ATP, which then is used for glucose. All right, so that's the order there. And what's the glucose going to be used for? And what other molecule or what other process makes ATP? And 30, hopefully you have cellular respiration. All right. And it's interesting, I had a handful of people in their drawing that I corrected last night. And their drawing had actually put a transport protein in the chloroplast. They actually put this on there and said that you need a transport protein so that glucose can leave the chloroplast. And where would glucose go in a plant cell to be used for energy? The mitochondria. It's going to go to the mitochondria where what's going to occur? Cellular respiration, and we come full circle. So, all right. I was pretty impressed that some people put that together on their own. I was impressed with that. All right, and the last little bit here, let me. This one. Was this picture here, and this was talking about some algae and doing photosynthesis, and what happens to, hold on with me for a few more minutes, let me get these answers done. The algae going through photosynthesis over time, what happens to these 3 molecules? So hopefully, without even looking at the graph, if algae was doing photosynthesis, hopefully you'll be able to tell me what would these three molecules would do. So CO2, if something's doing photosynthesis, is the CO2 that they have available going to go up or down? Down, because CO2 is used in photosynthesis. Hence, CO2 goes down. All right, so that's that one. Oxygen, if it's going through photosynthesis, the algae, oxygen is produced in photosynthesis. So if something is going through photosynthesis, the oxygen should go up, which is what we see here. So they're doing photosynthesis. So the algae is doing photosynthesis. The oxygen goes up. RUBP. Notice here the RUBP kind of doesn't really go overwhelmingly up or down, and that's because RUBP is here as part of the Calvin cycle. And so remember that it's used and becomes PGA first, but then it recycles and comes back to RUBP, so you never run out of it in the cell. So that's why we see a little bit of fluctuation, depends on where it's at in the Calvin cycle. All right? And then lastly, 32 gives you an equation, and this is the reduced equation. So A asks you to compare this equation to the real equation, and what they did is simplified it, they reduced it, so it's only showing you the net use of water. So instead of 12 waters going in and 6 waters going out, they're only showing you 6 waters going in. And so that's what it's showing you. And then B, why is this oversimplified? Because when you look at a reaction like this, it looks like if I just plucked out 6 CO2s from the atmosphere, took out some 6 waters, shine some light on it, I should magically get glucose, this reaction. And that doesn't really happen that way. You need a chloroplast, you need a bunch of enzymes, there's a lot of steps involved in there, and so that's why it's oversimplified. But it gives you the big picture as far as what goes in and what goes out. All right? And that's photosynthesis. All right. Let me shut off my video.